Hello, am I audible? Am I clearly audible to everyone? Is my screen visible to you all? Okay. So welcome to week 4 of this problem solving session. So those of you who are joining for the first time, I am Chandra Paul. I am a PhD scholar at Chemical Engineering Department here at IIT Kanpur. And I have been working in the field of tissue engineering for my thesis, because of which I have been also been exposed to various kind of biomaterials and fabrication various kind of 3D structures using these biomaterials. So if you have any queries during this session, feel free to raise your hand, okay, and ask directly by unmuting yourself. If other than that, if like after this session also you have additional queries, you can post it in the discussion forum. All right. So usually every week we meet on Saturdays from 4 to 6 p.m. and all the sessions are recorded and they are available in my YouTube channel. The notes are also made available to you by the NPTEL people. So in your portal, you won't be able to see the chat option. So if you have any queries, you have to unmute yourself and ask, okay? Or you can raise your hand as well so that I can call upon you, okay? So in last week, there was a query actually. Uh, there were few students who were facing bit difficulty in understanding the expression for creep, okay, or creep recovery phenomena. So I'll briefly discuss that and basically introduce you to a few additional topics that will better help you understand these two properties, the stress relaxation and creep recovery of viscoelastic materials, which are not covered in Professor Doble's lectures, but I found this content from a polymer science lecture, okay, of NPTEL only. And I'll like briefly tell you so that you will better understand the significance of those expressions that you used for solving those equations on stress relaxation and creep. So if you so we use a machine called rheometer, all right, to do to measure how the material or viscoelastic material is relaxing in presence of stress, that is stress relaxation we measure, or also we see how it is creeping, okay. And so you know what is stress relaxation. Here we keep the load. Here we keep the strain constant. So initially we raise the strain and then we keep the strain constant in the machine. And as a response, what we see that initially when the strain is increasing in the material, the stress also increases. All right. But when the strain is kept constant, the material, how it responds by decreasing its stress. That is the stress relaxation phenomena, where the strain is constant and the stress is reducing in response to it. Whereas creep recovery, so these are like simultaneous events that happens. So in the material, what we do, we increase the stress and we increase the stress to a constant level. All right. So in response to that, what we see in the material, the strain is increasing like this with respect to time. So all these phenomena is happening with respect to time. So when we are increasing the stress, the strain in the material is increasing, that is creep. But the moment we remove this stress, that is there's a drop in stress is there, then we will see the phenomena of recovery. And how the material strain is going to change, the strain will decline and the decline in strain will happen in this way as the trend in the graph is shown. And all this happen with respect to time. So these are two phenomena that are seen only in viscoelastic materials. Okay. So people use models, all right. There are two models. One is Maxwell model, another is White model to explain these two phenomena. All right. And the expressions that you saw in the lectures, those expression came when they were when we solved these two models. On solving the Maxwell model and the White model, we obtained those two major equations. All right. So I'll not go in too much detail about the models and the subsequent derivations and all. You can do it on your own. I have, like in my slide, I have given the reference as well. You can go to the NPTEL lecture, all right, by Professor Abhijit Deshpande. So he is teaching a course on polymers, and since polymers cover a very big class of viscoelastic materials, so there this kind of mathematical treatment is more important. But for our case, I'll just give you a slight primer, kind of like what is Maxwell model and White model, and what expression we obtain from each of these models. So I hope my screen is visible. All right. Right. So the Maxwell model is a model where the spring, which is elastic in nature, and the dashpot, which shows the viscous property, these two are in series. So this model also assumes that the stress in the spring and the stress in the dashboard, they are equal, whereas the strains, they are not equal. Because of this, we obtain some expression, all right, and on solving that expression, we get the expression for stress relaxation, where the strain is constant. And if you remember, this is the expression you solved or you use to obtain the relaxation time, correct? So this expression is obtained from the Maxwell model, all right. You see, this is exponential, all right, there is exponential term, where if you compare that exponential term here, there was e to the power minus some value, right? Negative. So in this region, when you are in this region, all right, the decrease in stress is happening exponentially and it is decaying. Thus, the negative sign is explained. Okay. Whereas Maxwell model also gives expression for creep, but this creep expression looks like this. If you plot this expression, then you get a creep graph that has a constant intercept and it is linearly increasing. So this type of behavior is not correct, or this representation of creep is not correct. The creep doesn't happen like this. If you if you see this particular curve, the increase in strain when you're applying some stress is happening like not in a straight line manner, all right? There is some non-linearity in it. So that is why Maxwell model cannot represent creep accurately. Then there is another model called Kelvin White model, or simple White model is also, it is also known as White model. So here you see the spring and the dashpot. So these are in parallel now. In Maxwell, they were in series, but here they are in parallel. And the assumption here is the strain experienced by the spring and the dashboard, they are equal, but the stress is not equal because of the configuration. They are in parallel because of that. So if you again solve the expression for this model, you will get a stress relaxation equation which looks like this. And if you plot it, where stress versus time, if you plot it, you get a straight line. So that is not correct, right? So for a constant strain, you do not see a time independent increase in, so time dependent the strain, the stress is not independent of time. Here it is not a constant value. So that is why Kelvin White model cannot represent the stress relaxation phenomena, but it does gives us the correct expression for creep. And if you remember, this is the expression that you have used for the problems where the material, the polymer piece was experiencing some creep behavior, and there you had to calculate the retardation time and then the a percentage change in strain, right? How the from the original strain, how much deviation is there with time, right? So in those problems, you use this as particular expression. And Here again, if you look at the curve here, you see the non-linear, the exponential behavior in this region as well, and in this region as well as in this region, can be explained by this component here. Okay, so these are the like fundamental explanation behind the expressions used in the lectures. Yes, anyone has any query? If not, you may unmute yourself. Sorry, mute yourself. Okay. So, as I was saying, that these two are the main equations that you used for solving equations on creep and stress relaxation. Okay, so don't go into too much detail into how the expression are obtained and all. But if you are interested, I suggest you to go to this lecture, lecture 48. Okay, by Professor Deshpande. It is an NPTEL course on polymers. You can have a look at it, but it's a bit more complex and out of scope for this course. But if you're still interested, you can go through this lecture. Uh, Otherwise, if there is any other query, you can let me know.
Hello, ma'am.
Yeah,
Uh, Ma'am, I have a doubt in the assignment question.
which assignment question?
Ma'am, week 3 assignment.
Week 3. Okay, all right.
Ma'am, in question 7 and in question 8, I have doubts. You remember you asked us a question on a contact angle, but it was in the terms of hydrophobicity,
Mm-hmm.
right? Actually, we got the same question in week 3, but there it was in the terms of hydrophilicity.
Hmm.
But they have accepted the answer for hydrophobicity answer and not the hydrophilic answer. Like, should I tell you the question if you want?
Actually, I have it in front of me. Question number 8, right? Contact
Yeah.
angle of a surface. So if you go to the forum, this query has already been asked there. The correct answer is actually A, option number A. All right. There is some. I've also informed the other TA here. If you have answered option number A, then it is correct. The current option that is given here, that is actually wrong. They will rectify it.
No, ma'am. In question 8, they have accepted A is more
Oh, sure. Okay.
hydrophilic than
Okay.
B.
Okay. Sorry. B is the correct then. Sorry. B is more hydrophilic. That is the correct answer. They will be rectifying it. I meant to say that. Yeah.
And ma'am, there is also a doubt in seventh question.
Mm-hmm.
Like, which surface coating does not cause killing of bacteria, and the accepted answer is antibiotic coated.
So I again discussed this question with the other TA. The correct answer is antibiotic coated only. All right. So what happens is silver ions, it itself has the antibacterial property. So if you coat the surface with silver ions, it will show antibacterial activity. Then people have also reported that nanostructure, like they pattern the surface at a nanoscale. So, and when they use those surface to grow bacteria, there they also observed antibacterial activity. So in literature, it is there. And enzyme immobilized means what kind of enzyme we are talking about means there can be those enzymes that act on the peptidoglycan layer of the bacteria and thereby weaken its defense mechanism. Okay, so if you are coating with those kind of enzymes, then it will cause antibacterial activity. Whereas simply uh, like bringing antibiotic molecules in contact with bacteria doesn't ensure that it will kill the bacteria. Most of the antibiotic has to be like internalized by the bacterial cell for it to act on, say, any cell wall component, cell wall component, or any gene level expression. So simply coating it. When you're coating it, it means you're bringing that molecule in contact. Okay, the bacteria is not internalizing it. So antibiotic is effective if it is in a like soluble form. Okay, it is in soluble form, so that the bacterial cell, inside the bacterial cell, it can easily enter and do its activity. So which is why option A is correct. Okay.
Okay, all right, ma'am.
Mhm. So no other query other than what you have, like she has asked, any other query? Okay. So I hope the equations on stress relaxation and creep recovery is pretty much clear now, and you won't get confused next time when you'll be applying them for solving more problems. So coming on to revising some of the concepts that were discussed in week 4. So we saw that there are some tools, all right, analytical tools that we use for characterizing biomaterials actually, to know which biomaterial is performing better for a particular application. So we can use various kind of techniques to understand the morphological properties, like how the surface is it rough or it is smooth. Then morphology of the surface, like we can actually image the surface. So for that we use techniques like SEM, or scanning electron microscope. Then to understand the roughness of the surface, we used AFM, or atomic force microscope. Then due to hydrophilic or hydrophobic nature of the biomaterial, it can cause water intake or water ingression. Okay, and because of which the whole material can swell, and indirectly it will affect the morphology of the entire material. So for that we calculate something called swelling ratio, which basically means how much the material weight has increased with respect to the dry weight of the material, means when the water was not present. So in that way we compare the change in the morphology of the material when the water has been taken up. Then we for biomaterials which are prone to degradation, means when they can undergo biodegradation. Okay, so for tissue engineering we mostly focus on those biodegradable biomaterials. So for characterizing, so to understand how fast the weight loss will be, okay, or what is the rate of degradation, we find out the weight loss maybe with respect to time or with respect to the reagent. So if it is hydrolysis based, or it is ion based or enzyme based, with respect to that, we understand how the loss in weight is happening. Okay. Then surface changes. So if you are, say, as I was saying, nano pattern, okay, so there are micro pattern. So that patterning changes the surface characteristics. Then it can change the hydrophobicity or hydrophilicity. Then, and for that we measure the contact angle, all right, or we use AFM to understand how rough it is. So these are surface changes and the techniques through which we understand how the surface is changed. Then mechanical properties, how the mechanical property of a material is changed with respect to, say, a continuous stress and strain is being subjected to the material in form of maybe compression or tension or bending. Then how the material is declining, how the performance of the material is declining. Okay, so that we characterize. Then the some of the polymers, especially not metals, not ceramics, but polymers, they can undergo changes in their molecular weight, overall molecular weight. So we calculate those molecular weight. So we can calculate the number average molecular weight average molecular weight, viscosity average molecular weight. So usually when we purchase a polymer for our application, some molecular weight is given by the manufacturer. But when we have made some kind of 3D structure out of it and using it for an application, then it doesn't mean that in that particular application the molecular weight will remain constant. All right. In that case, we have to determine it ourselves. Okay. Then these values are determined. How they're determined? They are determined using various techniques like GPC, gel permeation chromatography, where a larger macromolecule, if it is say degraded, all right, or if it is broken into smaller oligomers and all, then based on their size, it will permeate through gels of particular pore size. Okay, and then we get fractions out of it, and we can analyze those fractions using mass spectroscopy, etc. All right. Then we also understand how in a particular condition the thermal properties of a biomaterial is changed. So for that we use techniques like DSC, differential scanning calorimetry, then TGA. All right. Then the crystallinity of a particular material is also important. It determines indirectly whether it will degrade or how fast it will degrade or not. So how crystalline it is or whether it is amorphous. Okay, so these kind of answers can we can obtain from a technique called XRD. Then if we want to understand the surface chemistry, so there is surface property or surface morphology that we got to understand from measuring the contact angle and understanding surface roughness. Then there is something called surface chemistry, the functional groups, chemical functional groups that are present. All right, because those also have an effect on how the cells are going to interact with the biomaterial. So for understanding the changes in surface chemistry, we use techniques like Fourier transform, infrared spectroscopy, then UV based Raman, NMR. All right. So these techniques we used. Then in order to understand whether our material is biocompatible or not, all right, so we have to understand how whether it promotes bacterial biofilm formation or not, and if yes, what is the characteristic of this biofilm, then how cytotoxic it is, means whether it promotes cell death, and if yes, then how much is that percentage. Then if the biomaterial in brought in contact with blood when transplanted inside the body, and whether that biomaterial is capable of coagulating the blood. If yes, then it is very dangerous. All right. So these kind of characteristics we do, before we fully say with confidence that this biomaterial is suitable for this particular application. For example, tissue engineering. All right. So briefly, I'll discuss few of these techniques and together we will also solve few problems based on that. So first, broad umbrella of technique is the microscopy. All right. So the microscopy, you can again classify it based on how it is done, like what is the light source here, or how the uh, sample is illuminated. So if the sample is illuminated by visible light, all right, then it will be called light microscopy. All right. The sample is exposed to electron beam, and we are using electrons as a way to get a contrast image. All right, then it will come under the group of electron microscope. And then there is another group of microscopy that is called atomic force microscopy. Now this particular technique is not you cannot actually, it's a hybrid form of microscopy, you can say, because here you are basically using a cantilever probe to map the surface. It is moving across the entire surface of your sample, and how the probe is moving, whether it is moving up or down, the valleys and the peaks, how it is moving. Based on the movement of the cantilever, we get an image. All right. And that image tells us how rough or how smooth that particular surface is. So I have some images in front of you. I don't know. Can you guys confirm which slide number you are able to see? Is it slide number 10 or 11?
11.
11. All right, great. So in this slide, I'm trying to show here the first image is actually an image of a tissue, okay, taken from taken using light microscopy. All right, and light microscopy, in comparison to other techniques, is the provides a very like comparatively or relatively provides a lesser resolution, around 100 nanometer or zero point 0.1 micron, right? The second image here is the face of an insect that you got from scanning electron microscope, where the electron beam is limited on the surface. Only the surface is onto the surface, the electron beam is coming and deflected. All right. So using this technique, we can obtain image that looks something like this. So the surface features of that insect is very much prominent. And the resolution minimum that we can go here up to zero point 0.2 nanometer. Now, some electron microscope, some scanning electron microscopes also come with an additional attachment that is called EDX. All right. Now, using this attachment, we can also understand what all elements like carbon, nitrogen, oxygen, these are present on the surface of the sample, all right? Not in the bulk, but on the surface. So this is an additional information that we get about the surface of our biomaterial. Then TEM, also called the transmission electron microscope. Here, the electron beam, it basically transmits through the sample. And whatever electron beam we get from the other end, we collect it to get the information. So this particular image shows you that this is a TEM image, and it is image of nanoparticles, like which are of very small size, like few nanometers. All right. So and it is more appropriate for getting the images of nanoscale structures. All right. And the resolution is around zero point 0.5 nanometer, whereas the last image is actually taken using atomic force microscopy, where you are able to see how the surface is, the morphology of the surface, the roughness, all right? And this is again obtained from the movement of the cantilever probe. And the resolution that you can get is around 0.1 nanometer, right? So brief summary about all the four types of microscopy that we generally use to characterize a biomaterial. Now here's a question in front of you. So you have to match the following. So on one column, there is the name of the microscopes, microscopy techniques that we just saw, and their application, where we can use these. So maybe one of you can start think over it, and maybe one of you can start, like say A matches with which of the following four options in column 2. Similarly, B, C, D, like that. Go through the question first.
Ma'am, 12th slide is not visible for me. Is it same for everyone?
Okay. Now is it visible?
Yes. No. Yes.
Yes. All right. So go through the question, and whoever is able to understand, maybe you can start answering. So option A, scanning electron microscopy. Which of the options 1 to 4 does it match accurately? Or for which of the four applications can you use the technique scanning electron microscope, or SEM?
Third one, morphology.
Sorry, I couldn't understand. Can you repeat again?
It's the third one.
Third one. All right. So you're correct. We can use SEM to understand the morphology of cells on the surface of the biomaterial. Then option B, compound light microscope. So for which of these applications can you use 1, 2, or 4?
Fourth.
Correct. And option C, only two are left, 1 or 2. TEM can be used for which of these application? One or two?
First.
First, you're correct. And then we are left with D, which matches with option number 2. So all of you are correct. So is there any doubt in understanding why these options are correct from anyone? Okay, so I'll move forward. So next technique is called XRD, or X-ray powder diffraction. The powder here means that we are using sample in powder form. So this particular technique is very useful in understanding the phase of our material, whether it is crystalline, amorphous, okay? And we can also use this technique to determine the unit cell dimension. So if our material now, why this technique is important for understanding biomaterials. So if the biomaterial is made up of metals which are usually crystalline in nature, then we can say that the effect of the biological environment on degradation is comparatively less. But if it is, say, made up of amorphous materials like ceramics, then it has a higher tendency to degrade faster. So the crystallinity of the material has a huge impact whether it will degrade faster or slower. Okay. And this particular technique is based on a law called Bragg's law, all right, which is given by this expression. I hope you guys have seen in your school days. So here n is a positive integer, lambda is the wavelength of X-ray with which your which is incident on the sample. Then d is the distance between the lattice planes. All right, you can say the part difference. So there's a sample and it is having some unit cells and is forming a lattice. The distance between two lattice planes, like 3D planes, are there. And theta is the angle between the incident ray and the plane. Plane here is the sample. So on the sample, some light, that is X-ray, is incident. So this will be your theta, and it will then reflect. And when it reflects, it the nature of the sample determines the quality of this reflected light. Okay, means whether it is, and 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 we actually take it, this particular reflected light is taken by the detector of the machine, and it measures the uh, intensity of that particular reflected light. And type of the sample, for a particular type of the sample, like say crystalline sample, it will have a signature type of diffraction pattern. Okay. Similarly, for amorphous sample, you will get another kind of pattern, and we compare it with some standards. So there is a standard material which we know is crystalline, or it will give a peak, a high intense peak at this particular region. So we say, since this particular, my unknown sample has peaks at this region, and the known sample also has peaks in this region, that means my unknown sample is more towards crystalline form. In this way, we do comparison and come to conclusion whether our sample is crystalline or amorphous. So here's a question for you. The Bragg's equation, which we just saw, in that particular equation, what does theta represent? There are four options, only one of them is correct. So theta is angle between scatter plane and reflected ray, angle between scatter plane and incident ray, angle between scatter plane and lattice plane, angle between two scatter planes. Yes, someone has unmuted themselves. Would you like to answer?
Option B, ma'am.
Option B. Yes, you're correct. Okay. So option B is correct. Theta here signifies the angle between the incident ray and the scatter plane. Here scatter plane is basically our sample, all right, in powder form. Now there's another problem for you. So this problem is based on the unit cell. So how the unit cell is, whether it is cubic, tetragonal, rhombus, orthorhombus, etc. So um, this part is not taught in the lectures, all right? So maybe I'll first quickly give you a primer before you start solving this particular question. So um, what we do, we first draw the three axes x, y, and z. And there is a unit cell. The unit cell will have length, edge length A, B, C, and the angle between these edges is alpha, beta, and gamma. So the plane, which is XY plane, will have the plane with edge length A and B. All right. Whereas in the XZ plane, you will have a face of the unit cell with length C and B. Whereas how the angles are named, the angle between the edge B and C is called alpha. Angle between the edge A and B is called gamma. And beta is between the edge A and C. Okay. So in this question, we are given the length of the three edges of a unit cell and the angle between them. We have to find the longest diagonal, which is the longest diagonal, and what is the length of it. So if you, so let me draw this particular unit cell. Okay. So in this is just a representation. We can draw a unit cell with 90-degree angle. So it will look like a cube. So this is just a representation where alpha, beta, and gamma are not defined. But in this diagram which I'm drawing, I'm saying that alpha, beta, and gamma, they are all 90-degree. Okay, so I'm not drawing very nicely. Let me. So in this unit cell, I'm saying that alpha, beta, and gamma, they are all 90-degree. All right. So this will be Z, this will be X, this will be Y, and here A, B, and here is C. Okay. And we have to find out first the diagonals. What diagonals is possible? So one diagonal you can draw like this, which is in which can form a triangle like this. So this blue diagonal is called a face diagonal. Similarly, in all faces here also you can draw. All right. In the bottom also you can draw like this. Okay. On this face, then on top face, here also in back face. So diagonals which are in the face of this particular cube are called face diagonal. Then you can also have that what is called a body diagonal. So a body diagonal goes through the bulk of the body. So it will be like starting from one corner and ending at another corner which is opposite to it and on top of it. Similarly, there can be a body diagonal that can start from this point and end at this point. That is called a body diagonal. Now you tell me through maybe guess which diagonal will be the longest here, the face diagonal or a body diagonal? Like simple logic.
Body diagonal.
Yes, you're correct. So it is the body diagonal of a unit cell which is always longer than the phase diagonal. So if you are asked the length of the longest diagonal, you will find the length of the body diagonal. Got it? So how will we proceed? Now we will come to the question part now. So there is a unit cell, all right, which is having dimensions. So again, the 90-degree is there. So alpha, beta, gamma are all 90-degree. So that is why I'll draw it like this, not in inclined way or anything. Now A, so this is a B, this is A. They are equal, right? So for they are kind of equal. So it will be here 4 units, and C is the longest edge, it is 7. Okay, now you have to find out what is the length of the body diagonal. So you can take the diagonal which is coming from here, or you can take the diagonal which is coming from here. Okay, so whichever, both the diagonals will have the same length. So for my case, I'm taking a diagonal which starting from this particular corner ending at here, and I'm drawing. And to find the length of this particular body diagonal, I will use the Pythagoras theorem, which applies on right-handed triangle, right? So since the edges and all the corners, alpha, beta, gamma are all 90-degree, so this particular triangle which I've drawn in orange will also have a 90-degree. So I can simply use the Pythagoras theorem to find the lengths. So if I name the triangle, I can name it as X, Y, and Z. So we have the length YZ, which is 7 units, but we don't know the length of XZ and we don't know the length of XY. And XY is the answer, all right? XY is the answer that we need. So first we have to find XZ. Now, if you notice how the XZ is placed here, see, it is again part of a right-handed triangle whose uh, one edge is 4, another edge is 4, right? This is B and this is A. So XZ is a part of another right-handed triangle. So this particular corner is 90-degree. So for finding XZ, we will use the Pythagoras theorem. So XZ will come out to be root 32. Okay. Now we have XZ, just we need to find XY. So what will be XY? Use Pythagoras theorem. 32 plus 49, it is 81. Then XY will be root over 81, which is 9. Correct? So the answer here is 9. Any doubt in understanding this particular problem and how it is solved? No doubt. Have all of you understood the logic behind solving this kind of problem?
Yes, ma'am.
Okay. Now, in order to understand how the biomaterial, the thermal properties of the biomaterial is changing, okay, we use two main technique. One is called TGA, Thermogravimetric Analysis. Another is called DSC, Differential Scanning Calorimetry. Now, these two techniques gives two different information. So First technique TGA, it helps you understand how the weight of the material is changing with respect to temperature. Now, why this is important for us to know? Now, there are various kind of materials which are susceptible to high temperatures, okay? Or they have a tendency to lose weight at even uh, close to room temperature, okay, or just greater than room temperature. And if we are using a material which is susceptible to lose weight at 37 degrees Celsius, which is the temperature of our own body, then you have to understand how fast it is losing its weight. All right. So then again, you can understand the effect of our body temperature on loss in weight or degradation. So when you're interested in answering this kind of question, then the technique to use to get the data is TGM. Then DSC. So DSC is a technique which measures the amount of heat that you require to heat up the sample and increase its temperature. Now, if the required heat is in the positive direction, delta H is positive, then you know that exothermic or endothermic, you can categorize like that. And correspondingly, you can use this technique to determine the key temperatures of a polymer. What are those key temperatures? One is the melting point, another is the glass transition temperature. Now, these two temperatures are very important when you are working with polymers. TG, or glass transition temperature, is the temperature at which the material transitions from a glassy state, or you can say, polymers are never purely crystalline, they are always semi-crystalline, okay? So semi-crystalline is the best level of crystallinity you can get in a polymer. That's, and it transitions when the temperature is TG, or when temperature is above TG. It changes its state to a rubbery state, okay, or amorphous state. So at this particular temperature, the crystalline nature of the particular polymer has changed, right? Whereas Tm, here, what happens? The polymer changes from a solid phase to a liquid or molten phase. Okay. Now, tell me, which temperature will be greater? Will it Tm or will it Tg? Means here, which sign should I use? Greater than or lesser than? To correlate these two temperatures. Like guess, simple guess from whatever you have understood in the week's lecture.
Greater.
Right. So what happens when you obtain the graph from DSC, a thermogram it is called. We usually obtain, so here is temperature and here is amount of heat that you require. So we see that TG comes first and then TM. And after TM, the polymer is no longer a solid, all right? It is become completely liquid. And again, if you want to go back, you have to remove the heat from it, all right? And and then you will be able to reach a state which is semi-crystalline. So usually, and actually most cases, Tm is greater than Tg, or Tg is less than Tm. And the values of Tg and Tm is obtained from the technique called DSC, or Differential Scanning Calorimetry. So here's a question for you all. You have some polymer solid pellets, all right, and you want to dissolve it in a solvent. So at what temperature will you heat this mixture, solid pellets and solvent, this mixture of uh, solid pellets and solvent? so that you get a homogeneous solution. Will you heat it near melting point, or will you heat it near glass transition temperature, or will the heating will occur in between Tm and Tg, or you will just simply heat it at room temperature? Which of these four options seems correct to you?
Near TM, ma'am.
Yes, you're correct. So, can you explain to everyone why do you think, or why Tm, why you should heat near Tm? The logic behind your answer, can you explain to everyone?
As you said, we need to melt the solid to change to molten or liquid phase. It should be in TM range. So the temperature should be near TM, I
Yes,
guess.
yes, you're correct. Otherwise, you won't get a homogeneous solution. All right. You will get a phase separated kind of a thing. So you don't want that. So for homogeneity, you should melt near or at Tm to get a homogeneous solution of polymer and get a polymer solution. Now, there are various spectroscopic techniques that we use to understand our biomaterials. So there are some techniques. What is basically spectroscopy? Here we are using some form of say light, all right, electromagnetic radiation we use, or we use some electric field or magnetic field, all right. So in order to irradiate our sample, and upon irradiating our sample we get some information out of it, that is spectroscopy. And based on various other parameters we categorize them. Okay, so there is various kind of spectroscopy, but here I have listed only few which are relevant to us as a biomaterial researchers. All right, so the techniques which uses EM radiation, electromagnetic radiation, whether in the UV region or visible region or IR region. All right. So for, these four techniques are those spectroscopy techniques which uses EM radiation. What are those? First is atomic absorption spectroscopy. Then there is UV visible spectroscopy, IR spectroscopy, and NMR, or nuclear magnetic resonance spectroscopy. In all of these techniques, we irradiate our sample with with an EM radiation of particular wavelength. All right, and then we understand how its sample has absorbed it or emitted out the light, and then we get some information. Now, there is other forms of spectroscopy as well, where we first charge the sample, okay, the biomaterial sample, and the charged sample that interacts with the electric or a magnetic field. So this particular technique is called Mass Spectroscopy. Then there is another technique called fluorescence spectroscopy, where we uh, excite our sample in the EM radiation with using a wavelength of EM radiation within the EM spectra, and as a result we get something called fluorescence. All right, this fluorescence, the intensity of this fluorescence is usually lesser than the intensity of the incident light. So this again can be seen with a special class of molecules called fluorophore, or those molecules which can give fluorescence light. Not all molecules can give a fluorescence light. Then there is absorption spectroscopy as well, where some radiation uh, is absorbed, okay, and we get a characteristic peaks. So these all are spectroscopic techniques, and now I'll show you guys how they are used for answering various kind of questions in biomaterial research. First is fluorescence spectroscopy. I'll like briefly introduce. So this diagram is, maybe some of you have seen in your graduate level. This is called Jablonski diagram. Now this diagram helps you understand what is a fluorescence emission. All right. So in brief, there is an atom. All right. and within the atom there are ground state, okay, and there are some excited states. So when you are irradiating your sample with some light, this light or the photons from this light will be used to excite these electrons to that are present in the ground state, and they will move to a higher energy excited states. And in the excited states there are something called singlet excited states, and depending on the energy each of these depending on the photon energy each of these electrons has observed, it will move to different singlet excited state. Now two things can happen. It can come back to a lower excited state or a state of lower energy without irradiating any light, or it can come back in a way that it gives out some light as a result. So when the electrons from a higher an excited state comes to a lower excited state, here there is no irradiation. Now in this phenomena there is no irradiation. When we see fluorescence, when the electrons present in the lowest singlet excited state, they transitions to one of the states in the ground state, then only we can see fluorescence. All right. And the intensity of this fluorescence is always less than the intensity with which the intensity of the light with which you irradiate your sample initially. And you can actually quantify the fluorescence intensity using this particular expression, where I naught is the intensity of the excitation light, okay, and all other parameters is given in front of you. So this is just a brief kind of understanding of how the fluorescence happens. So this fluorescence spectroscopy uses this particular phenomena to get the fluorescence, all right, and give you a graph, okay, and with the x-axis you have the wavelength, and the y-axis you understand the intensity, how sharp the peak is. And then if the fluorescence you're getting in the visible region, all right, that is 400 to 700 nanometer, you also get some colors, and you can use those colored fluorescence light to get some information. And these kind of fluorophores which give some visible light fluorescence, they can be also used for biological applications, like for staining particular organelles in the cell, like nucleus or mitochondria or anything, okay. Or you can label it, yeah, using this fluorophore some carriers like nanoparticles etc, and deliver it inside the cell and see under microscope the location, the co-localization of the nanoparticle inside the cell. So these kind of imaging you can do using the molecules which gives out fluorescence, and how much the fluorescence intensity is, you can understand through fluorescence spectroscopy. Then there is UV vis spectroscopy. Here the sample absorbs and emits light in the UV and the visible region. So UV region is around 200 to 300 nanometer, and visible is around 400 to 700, and total the entire region is 200 to 800. So this particular technique is very important, it becomes very useful when you're studying, say, RNA or DNA molecules. So DNA or RNA, you can understand their concentration because these molecules absorbs UV light. So how much UV light they have absorbed. Based on that, you can understand the concentration. So this becomes very important when you're extracting RNA or DNA for maybe cloning purposes, or you're making cDNA, or you want to do gene expression studies. Similarly, there are some molecules, organic molecules or inorganic molecules, which absorbs light and emits light in the visible region. So they give some light which having some colors, and again you can use these chromophores for again biological staining purposes, all right, and use it to see under visible light microscope and stain your structures, different structure of a tissue or a cell with different colors and distinguish them. So again, the design of these molecules, how you will design it, all right, and what is the characteristics, how much, at which wavelength it is absorbing light, which wavelength it is emitting light. These all information you get from UV spectroscopy. This particular technique is based on the Beer-Lambert law, and it is given by this expression: A is equals to epsilon LC, where epsilon is a constant called absorptivity coefficient, L is the path length, and C is the concentration. So in case of DNA, as I was saying, you from the spectroscopy, you will understand the absorbance value. All right, you will get the absorbance value. All right, so the path length, it depends on the instrument. All right, it is a constant somewhat, and this is also a constant value. And when these are constant, you will anyway get it from the instrument or from the software. You will be able to calculate the C. So many of the softwares nowadays which comes with these UV spectroscopy, they directly give you the C value. You don't have to calculate manually the C value. All right, this equation becomes essential when you are trying to plot something called a calibration curve. Okay, when you calibration curve of some known sample. And then some unknown sample you give whose concentration you don't know into this calibration curve, and you calculate its concentration. So this Beer-Lambert's law is becomes helpful in your case for calculating the calibration curve. So I have given you one question which is visible in front of you, which is based on UV spectroscopy. So you are going to now use this spectroscopy technique for plotting a calibration curve of a chemical molecule. All right. And the known values that you have is the reading, absorbance reading. So 3 units you got for a concentration of 0.6 millimolar, whereas 7 units you got for 0.8 millimolar. Okay. Now you have to first plot the curve. All right, get an expression of the curve maybe, and from that expression you have to calculate, first thing, what is the concentration if the absorbance reading is this. Second thing is, what is the absorbance if the concentration is this. Okay, so if you are able, if you have understood the question, maybe you can start doing it yourself. Meanwhile, those who have not understood, you guys can see your screen as I'm doing it with you guys. Okay, so the known values is absorbance. So from the Beer-Lambert's law, first let me write that. We know this. So as I told you, epsilon and L are some constant. Okay. And we can, in general, we can represent this Beer-Lambert's law using this linear graph expression, which is y is equals to mx plus c, because you get a curve like this. As the concentration is increasing, its absorbance value also increases. This is a straight line curve, and the general expression of the straight line curve is y is equals to mx plus c. So in first case, so here y is nothing but a, representing a. M is the constant value, which is epsilon m. So let me write like that. Y is a, m is epsilon l, and here C is the intercept, and X is your concentration. Okay, let me write intercept with some other constant like I. I is your intercept, so that you don't get confused between concentration C and intercept C. Okay, so this is what we understand from y is equals to mx plus I. Now we will simply put these values into y is equals to mx plus I, and we'll get two expressions. The first one is 3 is equals to 0.6 m plus I. 7, the next one is 7 is equals to 0.8 m plus I. Now from these two equations, we have to find two unknowns, m and I. So how will we do that? we can simply subtract these two equations from each other. In this way, we can remove I and we'll get the value of M. If you do that, you will get something like this. Simplify it further, you will get 20. So M is equals to 20 you obtained. Okay. Now you have to obtain the value of I. So for doing that, you can use any of this expression where you can just fit the value of M and get the value of I. So this come out to be 12, which is nothing but minus 9. So I is minus 9. So what expression we obtained? We obtained y is equals to 20x minus 9. This is the equation of the straight line plot. Okay, equation of the straight line. If we plot the values, given values of absorbance and concentration. Now using this expression, we have to get the answers for A and B. Now in A part, what was given to us? We knew the value of absorbance. It is 4.3 unit. Means Y value is given 4.3. We have to find the value of X. So we have to just convert this expression that we obtained here in terms of X. So Y plus 9 by 20. Y is given. So put this value and use a calculator to obtain the value of X. What is the value of X? I got 0.665 millimolar. X is the value of the concentration that corresponds to the absorbance 4.3 units. This is the answer for part A. For part B, we are given with the concentration value. We have to find the absorbance. Concentration is 0.68 millimolar. So Y we have to find out. So Y is equals to 20. X we know. X is 0.68 millimolar minus 9. Again, you can use a calculator to solve this. So I got Y value of 4.6 units. So those of you who are doing it, did you also get the same values as me or anything different for part A and part B? Is there any like query in understanding this particular question? So the answer is correct, which I have obtained. Have you also obtained the same answers? There is one problem in this week's assignment, which is based on this, like it is similar to this actually. So if you've understood this particular question, you can definitely solve that one as well. Any query? If no, maybe you can give a thumbs up. Okay, great. So every one of you understood this particular question, then I'll move on. The next topic that I'll briefly discuss is about mass spectroscopy. So this particular technique, as the name suggests, will help you obtain the mass of the sample. Okay. Now, how do we obtain the mass? So we first charge it. Okay. We make the particles of our sample charged first. Then only it can be analyzed, and then further it can pass through the detector and give the value of the mass of the sample. So the sample can be in form of liquid or it can be in form of gas. So if you are using a liquid sample, then you will use a solvent to dissolve it, all right, and pass it through a liquid chromatography. Now, why liquid chromatography? So if you have a mixture of polymers, say, where different polymers have having different lengths, polymer length, all right, and you want to understand the distribution, basically how much of the polymer chain is having, say, 500 Dalton mass, or how much of is having 400 Dalton mass. You want to get a bell curve or a histogram like this, okay, with the x axis is your like the molecular weight, okay, and for doing that you want to get some fractions out of it. So these fractions you can obtain from liquid chromatography. Or if your sample is volatile, if it is in gas form, then you can use a gas chromatography as well. Now these fractions of samples, it is passed through ionizer. The role of ionizer is to make the sample charged. If it is not charged, then it won't be able to interact. It won't be able to interact with the field, magnetic field or electric field. Okay, so it has to be made charged. We can make the particles of the sample charged by using various techniques, by electrospinning or direct electron like bombarding on it, or we can use a matrix that indirectly charges our sample. So these are the various techniques that you've already observed in the lectures as well. Now once the sample is charged, you have to analyze it, all right. So here the analyzer composed of some magnetic and electric field. Now how the magnets or the electric field is created. So that depends on various kind of techniques like ion traps, TOF, right, quadrupoles. So they like interact with the charge samples and like allows the charge sample to collect on a detector. Okay, once it falls on a detector you will get some graph, and that graph will help you understand the distribution of mass of a sample. Okay, and basically that is your information that you were looking for when you started with this particular technique. So this is one question for you all. So you can use liquid chromatography as I told you. Now there are two types of liquid chromatography. One is normal phase, another is reverse phase. So here I'm asking what is a normal phase liquid chromatography? So option A, you use a polar stationary solvent and a non-polar solvent. Okay. Option B, use a polar stationary solvent and a polar solvent. Option C, you use a non-polar stationary phase and a polar solvent. So solvent here is the mobile phase, and stationary phase is the phase with which your sample is binding. Mobile phase is the phase through which that is elsewhere, that is like running. Okay, you dissolve your sample in the mobile phase, and depending on what is characteristics, it will bind on the stationary phase. So which of these is the correct answer?
A.
Option A. Let's see. Yes, you're correct. Option A is correct. Now, similarly, what will be then reverse phase liquid chromatography? So, normal phase was you were using a polar stationary phase, all right, and a non-polar solvent. And what will be the reverse of it then? Which of these is correct?
Non
Huh?
non-polar stationary phase and a polar solvent. C.
C, you're correct. So it is just opposite of it. So depending on your sample, okay, whether it is more, whether it has more affinity to bind with a polar stationary phase or a non-polar stationary phase, okay, depending on your sample, you will use the combination, required combination in your look in your liquid chromatography. Now, another question. In a time of flight mass spectrometer, means you are using time of flight mechanism to analyze. Okay, in your mass spectrometer, there is an ion of 400 Dalton and it reaches the detector in 1.5 minutes. Okay, so then proportionally you have to answer in how much time does an ion of 500 Dalton will reach. So the hint here is if the mass of the sample or the ion is large, it will take a longer time to reach the detector. If it is smaller, if it is a smaller mass, it will take a lesser time to reach. So simple logic from this, simple logic from this hint, do you think 500 kilodalton, if the time taken by 400 kilodalton, and this is the time taken by 500 kilodalton, which of this is greater? Like which symbol should I use? Less than or greater than to correlate these two values? Like simple logic. Then we'll do the calculation. Anyone? Understood the question? So you're using a mass time of flight mass spectrophotometer. Now the principle of time of flight relies on this hint actually. Okay, so the larger the ion is, the more time it will take to reach the detector. Okay, if the ion is small, it will take a lesser time. Now, in this question, you're given an ion of 400 kilodalton for 400 Dalton, but it reaches in 1.5 seconds. Now, I'm in this area, I'm asking how much time will a ion of 500 Dalton will take? Will it be greater than the time taken by 400 Dalton or will it less than that?
Greater.
Greater.
Ma'am,
Yes, it
but
will be greater.
inversely proportional.
So,
One should increase and the other one decreases, no, ma'am.
right. So, your molecular weight is increasing,
Okay,
right?
ma'am.
Huh?
Yeah.
So, okay, sorry, it will be lesser then, right?
Even
500,
in the week 4 session also, SARS it's the same, ma'am. But that I have doubt, so I asked, ma'am. No.
yeah, yeah. What? Can you
Even the
repeat?
SAR in class did the same, ma'am. When the molecule is higher, the time taken is also higher. Even the SAR said same, but it stated inversely proportional. That was my doubt.
No, no, you're correct. The doubt that you asked me is actually correct. So, the here 500 Dalton is larger, so it will take actually more time. Correct? Sorry, I got confused. So first, let me solve it. So let me solve this particular question, then it will help you understand. Okay.
Yeah.
Okay. So we are given, let me just erase this. So we are given an ion whose molecular weight was 400 Dalton. Okay. And the time it takes is 1.5 minutes. Okay. Now, if we do unitary method and ask you what is the time a ion of 1 Dalton will take? Simple. It will take 1.5 by 400 minute. Similarly, then how much time will 500 Dalton will take? You just multiply. And if you do that, so what is the answer you'll get? You'll get 1.875 minutes. So the time T400 was 1.5 minutes, correct? Now here the molecule was small, its molecular weight is less, all right? So it is taking less time. And T500, the molecular weight is higher, it is heavier, okay? So it is taking more time. Does this logic make sense? Forget about the hint. Does this logic make sense?
Yes, ma'am.
Yes. So what
Yeah.
is so what is written in the hint? It is written that time, if we just write, so if a molecule, and if we just correlate the hint with respect to what we have obtained, so a molecule which is of higher molecular weight, according to this hint, it will take less time, right? Time taken by the ion to reach the detector. So if the molecular weight is high, the time it will take, it should be less. And maybe I'm confused, sorry, but the logic that I've given you in while solving the question is actually correct. Maybe your doubt confused me, but the logic here, I think it should make sense. A heavier molecular weight ion, it will definitely take more time to reach in comparison to the one which is smaller, okay? So just maybe revisit the lectures again, it will help you. Otherwise, the logic that I'm saying is correct, but I'm 100% sure. Maybe some confusion has gone into my head. All right. Santosh, I think you asked that question. If you if it is still unclear to you, maybe you can ask again, because my this thing is still bit foggy in my head after you asked me.
So,
Yeah.
ma'am,
Yeah.
uh, in this question, if we go by your logic, it means the heavier the molecule, the more time it will take.
Yes, correct.
So it is directly proportional and not inversely, right?
Yes. Yes. That seems correct. Yeah.
Okay, so this hint that is written here is not correct.
No, ma'am, but in lecture also, the sir used to say this same hint.
Sir also gave you the same hint.
Yeah.
Yeah.
Yeah, yeah.
Mm.
But he said the same logic, ma'am, as you said. When more the molecular weight, the more times it take. He also said the same, ma'am.
Maybe the statement needs to be corrected then. The logic is correct. Means obviously a heavy person, if he falls, it will take more time that for the person to fall, right? Because the center of the g center of gravity, like more weight it is there in comparison of one. जैसे हवा अगर चलेगा तो हल्का person जो को जल्दी गिरने का उसका चांस रहेगा, and the same force is applied on a heavier person. So yeah, the logic is correct, the statements need to be corrected. um, Sorry, let me go back. It will be directly proportional, not inversely. Yeah. So, is there any other query on this particular aspect?
Ma'am, but in
Mhm.
assignment question, the same question arise, ma'am.
Now it is up to you what is correct to you. If like I have also seen the assignment, and based on the lectures, I have also marked it. But until I solve this question right now, it feels like option. And if you go back to the assignment, let me
It
just
is equal to molecular time, I guess, molecular weight.
yes. The question is question number, a second, question number 7. So is proportional to molecular weight. B should be the correct answer. Now let's see what is made correct, because this assignment is not yet solved. Okay, if then once it is evaluated, then maybe we can have more discussion on this particular topic. Okay.
Okay.
So the logic you got, right? Does anything
Yes, ma'am.
else, yeah, if anything else, like obviously whatever ion, if it is having a higher molecular weight, you have seen how I've solved the question. You saw the values, right? 500 kilodalton, it will definitely take more time, around 1.8 minutes, whereas smaller molecule it take 1.5 minutes. So it is directly proportional, not inversely proportional. I didn't think about it actually when I was like when I came across this question until now. So yeah. So now I'll briefly discuss the characterizations that we do when we are talking about bacterial cells or mammalian cells. Only these two types of cells we are more focused on when we talk about biomaterials, because either bacterial cells can cause infections, or it is the mammalian cells like the stem cells or other our primary cells, etc. They interact with the biomaterials. So if a surface or a biomaterial surface is more susceptible to bacterial cell growth, then we have to understand the quality of the bacterial cell growth, and whether it if it is forming biofilm, then what is the biofilm composition, etc. So we can understand the protein carbohydrate component by using UV vis, all right, or we can do spectroscopic techniques, color-based spectroscopic techniques like Bradford assay, Lowry method to determine the protein content of those biofilms. But this Bradford and Lowry method are very well established methods, and they are used for other applications as well. Okay, but the purpose is to understand the concentration of the protein in a solution. Then in biofilm also we have to understand the colonies. Okay, so whichever species is forming the biofilm under a particular condition, how many live colonies it is able to form. That is called CFU count, colony forming unit. So we can do in vitro level study or in vivo level study. Then we can also see within the biofilm how many cells are live or how many cells are dead. Similar thing we can do for mammalian cells. We can grow mammalian cells on the surface of a biomaterial and see the effect of our biomaterial on the viability of the cell, whether it is causing more cell death or if or more and more cell life. Okay, so we for testing, we actually call it as live or live dead assay, live dead assay. So for that, we use specific kind of dyes, and these dyes are basically fluorophore dyes. They give out some fluorescence emission. So we stain these the biofilms or the mammalian cells with these dyes. Now, why two dyes? One dye is able to penetrate the live cells only, whereas another dye is penetrate only the dead cells, not live cells. Okay, and these two dyes give two different colors. So CyTO9, this is one dye. There is other dye as well, FDA. So these dyes give a green color fluorescence, which means these dyes are indicating the live cells. So live cells are stained green by these type of dyes because they're able to permealize within the live cells. Then there is another dye that is called propium iodide, PI, propydium iodide. So propydium iodide is penetrate the dead cells whose cell membrane has been damaged. Okay, so only the dead cells get stained by PI, and it gives a signature red color fluorescence under microscope. So in this way, we can actually visualize the distribution of live and dead cell within a biofilm, or the live and dead mammalian cells over the surface of or within the bulk of a biomaterial. Okay, then if we want to understand whether our biomaterial has some antibacterial activity. Then we can do additional tests like zone of inhibition test, okay, where we can identify like how much of the concentration of our biomaterial, or a particular nanoparticle or whatever it is which has some antibacterial activity, how much of that concentration is causing killing in the uh, bacterial cells. So larger the diameter of the zone, so we usually get a zone in circular form. Okay, so we soak a disc of, we cut out a disc of our biomaterial, okay, and keep it in center of a LB agar plate and allow the bacterial cells to grow. So if it has some antibacterial activity, we'll see that a circular zone is formed around it, and that zone is free of bacteria. There is no bacterial growth. Okay, and larger this the diameter of this zone, the more we say this particular biomaterial or whatever formulation is having higher antibacterial activity. Okay, and similarly, in presence of this antibacterial biomaterial, we can see how many live colonies are formed. Okay, if the number is less, then again antibacterial activity is high. If the number is high, then antibacterial activity is less. So in this way, we can answer many questions based on bacterial activity or based on the proper biological property of our biomaterial. Then these assays becomes important when we are working with mammalian cells, because after all, for tissue engineers, it is important that our biomaterial should interact very well with our cells, because after all, we are interested in making artificial tissues, but the cells is an important component. All right, so we assess the metabolic activity of the cell. The metabolic, if the cell is doing metabolism, it also indicates that the cell is viable. Then it also indicates that the cell is proliferating very well. As the metabolic activity increases, it directly proportions to the higher proliferation rate. So simple, only determining the metabolic activity of the cell gives us much more related information about the uh, compatibility, biocompatibility of a biomaterial. So how we do that, how we determine cell metabolic activity, we use assays, calorimetric assays. So these are two mentioned here, MTT assay and MTS assay. Here in MTT assay, we use a salt, tetrazolium bromide. The long name is there, but tetrazolium bromide. Here also MTS also we use another type of salt. Now both of these salts are able to can react with the mitochondrial enzymes. So in highly metabolic cells, the activity of those mitochondrial enzyme is high. Okay, so these enzyme, when the concentration of this enzyme is also high, so more of these enzymes comes and interact or react with this particular salt, and as a result we get an insoluble salt that is called formazine. And this salt is color, it having some purple color, and we have to solubilize it another solvent called DMSO, and then we use a UV spectrophotometer to give out uh, a peak, UV based peak, and we get maximum absorption at around 570 nanometer. And based on this curve, we can get the percentage cell viability, means how much of the percentage of the cells are well live. Okay, then we can also understand the proliferation. If we do MTT assay over a period of time, then we can understand how fast the cell is viability, because as the time passes, the cell number increases and the metabolic activity increases. So with time, we can, if we do MTT assay, then we can understand the cell proliferation rate. So this is a very powerful technique, and it is reported in almost all type of journals who and all type of papers where they are talking about the biocompatibility of their biomaterial with respect to mammalian cells, growth of mammalian cells. Then if it is not it is not desired that a biomaterial should cause necrosis. Okay, because after all you're using that biomaterial to promote cell growth. Okay, so that it helps in regeneration inside the body. But if suppose it does, we have to assess it as well. So to assess the necrosis, necrosis means the cell has basically first apoptosis has happened, and then it has completely fragmented. Okay, so it is called necrosis. So tissue also undergoes necrosis. It becomes completely सड़ जाना जिसको बोलते हैं ना, so that is like necrotic tissue looks like, becomes completely purple and becomes darkish in color. Okay, no oxygen there. So that thing happens at cellular level as well. Now, how we assess that? We assess by using an enzyme called lactate dehydrogenase. Now, this enzyme is basically present in all the cells, but when the cell is necrotic, or when the cell membrane is compromised, like in cell apoptosis, this enzyme comes out. Okay, and in this assay, we basically understand the level of this lactate dehydrogenase. So the higher level of lactate dehydrogenase, it means the cells, more and more cells are towards cell death, okay, or necrotic phase. And we can also assess how the cells, or whether cells are adhered on the surface or not, how the cell has spread, means what is the final morphology. So we can assess it using SEM, or we can do staining as well, where we stain the cytoskeleton of the cell and the nuclei in order to get very nice colorful pictures of single cells. Or and in this way, so we can do SEM, as well as another technique, let me write. We can also use fluorescence microscope, where we use some colorful stains, fluorescence stains. Then other things that we assess are cell migration. So there are various ways to understand whether on our biomaterial surface the cells are able to migrate or not. So we can use transwell systems. Okay, transwell uh, systems. So cell migrations always happen in response to concentration gradient of a chemotactic agent. Of a chemotactic agent. So these chemotactic agents are nothing but some small molecular weight protein molecules which are released by a signaling cell, and these molecules, it diffuses. As it diffuses passively, it creates a concentration gradient, and the receiving cell, whichever it binds to, it responds to it. This chemotactic agent, it binds to the receptor on the receiving cell. All right, it gets signaled, and it starts migrating towards the source from where the chemotactic agent was released, that is towards the uh, signaling cell. So it is able to kind of sense the gradient. If the gradient is not there, the cell migration will not happen. So we, in transwell system, what we do, we use a well plate. This is one well, all right, and we use another well like this. Now the surface of the well is basically a membrane, all right. What we can do, we can fill the bottom well, a bottom compartment, with some liquid and a chemotactic agent like this, and the cells can be seated on the surface of the transwell. This is called a transwell. Now, on the transwell, there is no chemotactic agent, but in the bottom well, there is. So, since the surface is porous, and we keep the porosity such a way that the cells can like squeeze themselves and migrate, so it will start migrating, and after some time, you will see few of the cells have attached to the bottom. So, we can use this kind of system to study cell migration in presence of a concentration gradient. Or you can, what you can do, this is a 3D system. Okay, you can also do it on a 2D system. You can take a glass slide, all right, and coat it with some protein that allows cell adhesion. So you have adhered the cells on this glass slide. A confluent, very well tightly packed cells will be present, And what you do, you create a scratch like this with a pipette tip or something sharp, okay, which removes a chunk of cells from the center of this monolayer. Okay, second is called scratch test. And then, in presence of some media, proper oxygen condition and temperature, etc., you allow the remaining of the cells to grow. So what you will see that slowly over a period of time, if you click images under light microscope, you will see this particular region which you cleared it by making a scratch, that is getting filled up by the cells from both the edges. And then you can say that this filling up of this edge, this particular area was filled up because the cells migrated. There is no other way the cells could reach this particular region because you didn't seed any cells after scratching it. So this is another way to study whether the cells are able to migrate or not. Then people also study cell differentiation. So this becomes important when you are using stem cells for your application and you want to see whether your biomaterial is promoting any changes in the stem characteristics, means whether it is promoting its differentiation to a much more mature state, like some bone cell or a cartilage cell or something like that. So for understanding that, we do some tests like gene expression studies we can do, or flow cytometry studies. For gene expression study, we rely on the mRNAs. We collect all the mRNAs synthesized within the cell, okay, and do and understand which of these mRNAs concentration is high, and say that this particular gene is upregulated, and say that since the signature of the gene is this, and the signature of the gene expression corresponds to a mature cell type. Whereas in flow cytometry, we check the surface protein expression of surface proteins. Okay, if the surface protein uh, of a mature cell type is expressed higher, okay, and that is not expressed in the stem cell, then we say again that the cell has differentiated to this particular cell type. And other than that, we do for apoptosis, there is another test where we study the DNA fragmentation. So this basically gives you a small understanding of all the biological characterization, but there are various others people are reporting nowadays in the literature. So based on what you want to answer, like what is your question, you have to dig out and you will find many such characterization which you can do yourself and answer your questions. Okay. So there are a few questions based on what we have discussed so far. So first question is this: the release of the enzyme lactate dehydrogenase from a cell indicates what? Does it indicate cell apoptosis, cell attachment? Attachment means cell adhesion, cell differentiation, or cell necrosis?
Cell necrosis.
Yes, you're correct. Similarly, another very simple question. MTT assay is performed to study what? Cell attachment, cell metabolic activity, cell cycle, cell migration.
Cell migration.
And no, that is not correct. Anyone else? MTT assay you use to understand what?
B.
B. You're correct. Okay. So that's it from my side in today's lecture. Okay, and I hope whatever I have discussed you will be able to use and solve this week's assignment. If you guys have any queries related to what we have discussed in what I have discussed with you, you can ask me. If you have other questions as well, also you can ask me. Okay. Now I have enough time to do a formal or informal discussion. Any queries? So last week I discussed FTIR, all right, which is why I skipped in this week. So if any one of you have difficulty in understanding FTIR, then maybe you can ask, or any other technique that's discussed and you didn't understand. Whatever I have discussed today, is it clear to everyone? Is there any doubt in anyone's mind regarding anything? So the time of flight, one that question, is it clear? As the mass increases, the time it will take to travel, it will be greater,
Yes, ma'am.
right? Okay. So there is no queries, then maybe we can wrap up today's session. All right. I'll see you guys next week on Saturday, where we'll discuss some of the topics of week 5. Meanwhile, try solving week 4 assignment and submit it. So if there is any queries after solving, then also we will take up next week. Okay, then. Thank you all for joining. You guys can leave the meeting now.